Well, thanks for all of you guys. So welcome to the afternoon section. So now we switch to the Sue's Connect to share about some of the exciting things at Iowa State and Poultry Farm.
Thank you all for sticking around after lunch. Hopefully I can keep this at least light enough that everybody doesn't fall asleep. I do it at 8 o'clock-ish in the morning with some physiology students, so I'm hoping I can keep my streak going today. Unlike most of the other speakers, I did not have the fortunate opportunity to actually be one of Sue's graduate students or postdocs, but she was unfortunate in having to serve on my committee, which I greatly appreciated. She hopefully learned a little bit about dairy cattle, didn't catch my lies that I wrote in there, my typo. So it's been a pleasure to work with Sue kind of coming back at Iowa State on the poultry side. And so today, one of the things that I want to talk about is some of the probably a different legacy that Sue has helped to leave at Iowa State, which is the development of the farm. So this title is entitled, Gobbling Up New Opportunities for Excellent Phenotypes at the Iowa State Poultry Farm. So this image might be very familiar to Sue and some of those who were her graduate students up until about 2020. So this is actually a picture of the Iowa State Poultry Farm and the 7 buildings that we had at the time where we did all of our research, whether it was turkey research or laying hen research or genetic research. These barns were built, I'm going to say in the 1950s, around that time. And so by the early 2000s, they were becoming just, I'm going to say, slightly dated compared to some of the technologies that we have today. And so through Sue's efforts and many, many others, one of the things that happened recently is we were able to expand and modernize our poultry facility. So this has given those that work at this site a lot of opportunities and a lot of advantages that were not previously there. So one of the things I want to highlight is our Robert T. Hamilton Poultry Teaching and Research Facility. This is our laying hen facility where we house commercial hens, as well as the Highline Genetics Wing, where we have the genetics lines. We also have another facility, the Stanley Balloon Turkey Teaching and Research Facility. This is the second facility to come on site, and I should actually take a moment and step back and say that the Robert T. Hamilton facility was opened March of 2020. Actually, March 6th, we had dedication. It was a great and grand affair. We were able to actually hang out and interact with people. We placed birds March 17th. And the reason I remember this is I got a phone call from Panera going, so the governor has put in a ban of no more than 10 people. You have 12 box lunches or 20, I can't remember how many. Are you still going to need them? Because we'd like to know. And I said, well, yes, we still have to place birds. So it was a grand time to open that facility. And unfortunately, kind of since then, we've had a lot of things that popped up, whether it was COVID or high path avian influenza, that haven't allowed us to explore this facility as much with the public, although we have public spaces, and I'll get to those in a moment. With the Stanley Balloon Turkey Teaching and Research Facility, this was a facility that came on site shortly thereafter, and we opened that one in November of 2022. So it's been on site for just operational for just less than 2 years. And having been on building committees with Sue, we know that if you've been on one, you know they take a lot of patience, a lot of properly worded suggestions, potentially a lot of conversations outside, and some of those may involve alcohol or not. And so these are ways that maybe I didn't get to learn how to be a professional from Sue as a student, but definitely in those building committees I did. So thank you very much for demonstrating professionalism in some of those meetings. So I want to take the opportunity at this point to actually just kind of provide you with some of the modernizations that we've had with this facility and some of the unique phenotypes that we can gather with that. So this is an outline of the Robert T. Hamilton Poultry Teaching and Research Facility. And the first thing that I actually want to start out with is the teaching part. Within this facility, we actually have a classroom space, which is kind of unique because we can have this space where students can engage with birds. So up in the kind of the center top, so at 12 o'clock of your position, there's a group of students that actually rear their own broilers for 6 weeks during a class that Dr. Bobeck teaches. So we get to have that hands-on experience with students. And then we also can bring in speakers to this space and let them engage and see what it's like to be part of the laying hen industry, as well as that broiler industry. So these are great opportunities to grow the next generation, which we have been hearing a lot about of how important Sue was in on that aspect. So we want to continue that tradition with this area of the barn. The next area of the barn is the highline genetics wing. And this is where the very impressive, very important intercross lines are, the ones that have spurred countless numbers of master's and PhD theses and postdoc research. So we continue to house those lines there. And I'm hoping next year, kind of looking here for now, you have maybe some free time, 100-year-old birthday party, right? Because there's a line that is 99 years old this year. So I'm going to knock on wood that we can continue and hopefully celebrate in a year that they are turning 100 years old. And as you've heard today, there's countless opportunities and research that has come from these. Well, we'll see if it actually popped. There we go. So they were supposed to come across a little bit slower to show and highlight some of that research that Dr. Lamont's group has done. I focused here on the major histocompatibility complex, but you've heard lots and lots of research about different areas on the immune side that these lines have actually contributed to. So it's an impressive amount of work, and it is a little intimidating coming into that as a colleague now to fill this gap. So the next area that I want to highlight, apparently animation is not my friend today, so I apologize in advance, is the commercial laying side. So some of the work here allows us to look at various aspects, whether it's looking at conventional cage hens, cage-free hens, or our broiler systems. And so when we first moved into this facility, one of the things I really wanted to do is actually take advantage of some of these commercial lines and look at, can we develop some novel traits to look at intestinal health? And so as we think about how the cage-free bird and the conventionally caged bird interact with their environment, there's a lot of differences. One being that they can actually interact with their excreta. So these cage-free birds may be having and picking up different types of microbes, and immune cells are being activated. So one of the questions I had was, are we actually training the bird to develop their immune system? So we're doing some immune training with these different microbes that they're interacting with based on their housing environment. And ultimately, one day down the road, does that change based on what genetics they have? So why is this maybe important right now? In a lot of our industries, particularly the poultry industries, we're seeing that there's pressures to move away from the use of antibiotics. Whether it is for treatments, it's still okay, but we don't want to use them prophylactically. And so we see a large push for different probiotics, prebiotics, the no antibiotics ever. So we have breeding flocks that are not exposed to or given antibiotics, down to the broilers that are being marketed that also have never received antibiotics. So there's a large push to move away from this. So the idea of immune training based on the environment provides us with a gap, at least, that we can start to try and understand what's going on with the bird. So as we see this change from cage-free, from our conventional cage into our cage-free, then kind of this spurred the question along in our group of, do we see this immune training? Are we seeing differences in the microbial population? And if so, kind of what can we start to learn from that? So within this group or this project, one of the things we did was take intestinal samples to where we were running 16S, our RNA sequencing, to start to understand what types of populations were around. So of course, we started actually out with a big the big bad ones, right? The pathogenic microbes. And within this, we actually did not see any pathogenic microbes being upregulated based on housing systems. So from the standpoint of kind of our overall community, this sounds great, right? We're not exposing them to something that can cause disease. And when we randomly selected our birds, those birds did not have a disease pathogen. So that's great to know. But then as we started looking through the microorganisms or the OTUs that popped up, so these are single either clusters of OTUs that go back to a single organism or OTU, so operational taxonomic units, which are single DNA reads. What we started to notice was we could see favorable microorganisms. So in this case, our Candidaeus arthromitus and our Lactobacillus acidophilus were those that were favorable. Within these, we actually started to see that they were more favorable in our conventional. We saw that some of the potentially more detrimental, and I say that kind of with a question mark because they are either known as secondary pathogens, meaning that there has to be a stressor there for them to actually cause a concern or issue. So our Clostridium and our Galibacterium antineus, we're starting to show up, and we see those a little bit more prevalent in that cage-free environment. But what we really saw quite a bit of were these boxes in black, or this box in black. So the Lactobacillus kitsonia is one where we actually saw, well, some of the phenotypes were favorable, some of the phenotypes were not favorable. So how do we interpret that data? What do we do with that data, right? And so that's one of those things where we kind of sat and pondered for a while, and you find a paper that says this, you find a paper that says that. So we also decided that, well, 18S is one way of looking at it. Let's look at, or 16S is one way of looking at it. So we looked at 18S as well. That database is a little less populated. So what we saw a lot of were actually just yeast. So our Catsonia and Saccharomyces are all relatively grouped together in terms of a yeast family. But one of the more interesting things is this orange box, which would indicate Eimeria. And so it wasn't surprising necessarily that we saw a bit more Eimeria in our cage-free compared to conventional. So Eimeria is the organism that is associated with coccidiosis. So we might see more cycling of those oocysts, although it's at one of those points where you have to take a step back and remember that this is just DNA sequence. It doesn't tell us if these organisms are functional, if they're alive, or if they're just kind of present. So again, we see a lot of this data where we see single groups of either organisms or OTUs, but the bigger question kind of comes into play of what are they doing? What do they really do with the animal, and how do we interpret some of this data, and what are their outcomes? And so this is where we started asking the questions. Is it just a single OTU that is driving this? In terms of, like, if we see Eimeria, how bad is just seeing Eimeria sequence? And so one of the things we started to do, and this was with Kyron Anderson, one of Stefan Smith-Esser's grad students, is starting to look for correlations. So do we see hubs? Do we see a single OTU that might be driving more of these changes compared to others? And so we looked at this across both our 16S and our 18S sequences, so that we could start to get a full understanding of what those communities look like. So we looked at both positive and negative correlations, and we looked at them by housing system. So our conventional cage housing system, and then our cage-free. So it's really kind of probably a little hard to see here, but with our positive correlations, we actually see fairly similar results between both of the systems in terms of the number of connections that we actually see. But you might be able to make out that our conventional cage, we see a lot more negative correlations compared to our cage-free. Now, I don't know exactly how we want to interpret all this, and we need to take that step forward of looking at what do these individual communities look like. So one of the communities that I want to highlight is actually Eimeria. So Eimeria is where this little line is. And as we look from the positive correlations on this side, again, it's probably very hard to see. We actually do not see any connections. So we might have one connection, but there's no connections necessarily that are favorable. So when you have higher Eimeria, you're not seeing necessarily higher other organisms. However, we do see that when we have Eimeria, it could be potentially downregulating some of the others. So it is negatively correlated with other microorganisms. And so now it's a question of who are they and why are they downregulated? Are these cause and effects? Because we're looking at correlations, right? Number one thing you learn as a grad student, correlations are not causations, but it's just a question of where do we take this data from here, and how do we get more phenotypes that start to tell us who the bacteria or pathogens are that we need to focus on, right? If you look at feed additive companies, one of the things that they're always pushing, probiotics, prebiotics, symbiotics, and they're coming up with new names for them all the time. So how do we get the best one? And on top of that, does the bird control more of it than we want to give them credit for? So additionally, outside of just looking at who's present and who's present in what shifts and directions, right? Most of what we're looking at doesn't have phenotypes associated with it. We're looking at OTUs sometimes that have been discovered in the last 3 years in mice. And they're like, oh, look, it's here. We found it, but we don't know what it does. So one of the other aspects of this study was to try to understand what are some of the impacts that these microorganisms might have on the intestine. So one of the features that we looked at was intestinal permeability, which is the ability of a macromolecule. So we use a fluorescent isothiocyanate dextrin molecule, and we gavage the bird, and if it passes through, it's going to pass through between the enterocytes, meaning that it's a gut leakage marker. So typically we think gut leakage is bad, right? And so what organisms are we associated with more of this gut leakage compared to others? And so we did a lot of correlations, a lot more correlations. Probably what SAS thought I was doing every day for a long time was like, do you have any other thing you can do? So we wanted to look to see what are some of these OTUs potentially influencing in terms of who's associated with what. And so one of the things I want to highlight, again, not necessarily that I expect you to be able to read this, is that we had negative correlations with several lactobacillus bacteria. So lactobacillus, you see it a lot in your yogurts, right? So this is a lactic acid producing bacterium. And we saw several lactobacillus that were increased when intestinal permeability or leakage was down. So it might indicate that they're favorable. Sounds reasonable, right? Like, yay, that yogurt I've been eating is actually helping my gastrointestinal tract, maybe. But unfortunately, we also saw several that were the other way around as well. So we saw a lot of lactobacillus that were increased in OT in abundance and also increased with intestinal permeability. So this gets us back to, we need to know who's doing what and why. And so with that, I think, at least from the laying hen side, one of the things that we are looking into is how do these different environmental factors, health factors, age, as well as the host itself, impact what microorganisms are present and then what they actually do. So what is their phenotype once they are present? So used to class, I want to be like, so are there questions now? But we'll whirl into turkeys unless you guys have questions, and I'm happy to take them ahead of time. Okay. So with the Stanley Balloon Turkey Teaching and Research Facility, again, this was a building that was completed in 2022. And so with this, I want to kind of walk you a little bit through the facilities because we have some unique opportunities here in some of the data characteristics or data collection that we can do. So for those unfamiliar with turkeys, turkeys are a little bit more like the swine industry, where we start out with a brooder phase, where birds are reared up until 5 weeks of age. and then they're moved over to a grower facility for the remaining 15 weeks in a tom system, which we at Iowa State raise toms. So we have it set up to where within each of our 2 rooms of the brooders, we have 16 pens. And so we get to raise little guys like this. Sometimes they're a little bit more of an escape artist than we would like them to be. But for here, they get to live out 5 weeks of their life, have free access to feed and water. And so this is kind of a longer down the alley shot here. But what I want to highlight are the ability or the features we have of this barn. So the first is along the wall, you'll notice that we have 4 white lines. These are actually 4 different water lines that allow us to give up to 4 different water treatments per pen. So one of the unique things about this facility is we can change what one pen gets and the pen right next to it gets in terms of a water treatment, whether that's a vaccine treatment or a nutritional treatment or even microbial treatment. So we could, in theory, look at a lot of different variety of how water or water treatment impacts various aspects of the bird. The next, which is really, really hard to see, but one of the things that I am very excited about with this facility, especially from the collection of various phenotypes. So you might be able to see this little tiny gray box. Above every pen, we actually have data ports. We have up to 4 data ports per pen, which means we can collect environmental data at the pen level. We can collect video data at the pen level. We can collect audio data at the pen level. And probably a lot of things that I'm not even thinking of at this moment. So just thinking from that standpoint of what types of phenotypes can we collect? We have several. So that is something that makes me very, very excited. The other thing, we have these sensors throughout the entire barn. And part of this is it actually feeds back into our controller system. With the controller system, what's great about this is it actually utilizes artificial intelligence to maximize what the barn settings should be. So it will try to maintain your environmental settings through a variety of different means that I'm not smart enough to actually know. Brett is the one that Dr. Ramirez is doing a fabulous job of helping us understand that. But it also means that we have minute by minute updates of what's going on in the barn in terms of environmental output. So these are really cool characteristics that we have in the brooder. And then as we move these birds over into the grower, you'll notice one thing, it gets bigger. The birds get bigger, so they need more space. And so here we move them into a total of 64 pens. So we have 32 per room, but a lot of the same features are still there. But before we do that, in the brooder, we can hand feed birds. So anybody who's run a broiler trial, that's kind of about where we're at, where you will feed, a bucket of feed every day or so. But by the time you get to a 50-pound tom, you don't necessarily want to walk in with the feed. They do like their feed and they get a little aggressive. It is interesting to see how much they love the Lowe's buckets. So I think if you walked down the barn right now with a blue Lowe's bucket, you would become a friend. So one of the ways to not burn out grad students and our undergrads is that we have an automated feeding system. And so this is actually a picture of that automated feeding system that will dispense meals to each of the pens. And so as we look down and at our pens, these Y's across the top are actually what funnel the feed as it dispenses in each pen, its weighted amount into the feeders. So these birds have the ability to at least have feed intake at the level of the pen. We tried for feed intake at the level of the bird, but apparently that was not in the budget. They told me that was, a couple $100,000 more. So we work with what we but again, we have our data ports that I am very, very excited about. So now we can monitor those birds from the time that they're placed all the way until the time that we load them out for processing. So think about all the data and phenotype that we can collect with the right sensors. whether it's we're monitoring the bird for what is their aggressive behavior, because maybe that's something we'd prefer not to see. Maybe it's trying to understand what are the early cues of how these birds interact with each other, or when something such as lameness starts. And then there's kind of two things in this with this last arrow, probably very hard to recognize, but we also again have our barn sensors, so you can see the little red thing. But if you strain really, really hard, I didn't realize it was in the picture until after I was putting the arrows in, we have a scale. And it's one of our, one thing that we're very, very excited about is being able to walk toms on a scale. So we actually take the scale to the birds and walk them on. They're not like pigs, guys. They don't walk on. Looking at you, Derek. Derek's like, oh yeah, they just walk on and they stand there. And I'm like, we're not friends right now. Because we take the scale to them and we go, please walk on. And they go, no. And I'm like, please, please, I'm with a little help. And so this is one of the things that has made our jobs a lot easier, because we tried walking them to the scale like you might do pigs. That did not work well. So if you want to know how not to do that, we've got some answers to that as well. But within this facility, we're really, really excited about all the opportunities. Again, it's been open for less than 2 years. Most of our work now has been focusing on some of the nutritional aspects and questions that the industry has, but subtly trying to collect data, more and more data, so we can start to look at some of the genetic underpinnings that are going on in the turkey industry. So one of those I'm actually collaborating with Dr. Steibel on is trying to understand, they'll move eventually, I promise. As these birds grow, maybe. They're moving perfectly on my screen. Oh, there they go. They're very fast, if you notice. We'll see about the next video. So as they get a little older, they have different sounds. Well, what I'm trying to demonstrate with this is that they have a little bit of a different sound. They also have a bit of a different behavior. So and I'll try to see if I can get the last video, because I think the sound will also be very different, even if you can't hear them. Is that as these animals age, they change their behavior. So what you're missing out on is a tom fight at the end. Right? So these birds will have very different behaviors. And so just trying to understand, can we capture some of this data so that we can start to understand some of the early signs of maybe an aggressive tom or an aggressive environment that can be changed through management. Our birds here are at the commercial level. So one of the ideas is that we're actually keeping practical solutions to come back to the producer. So this is probably not going to work, but we have been collecting some of that data and are engaging with some of our undergrad population, because they are very interested in behavior and well-being, to capture some of these behaviors so that we can train computer algorithms to start collecting these at a higher rate than maybe what we can get a student with a camera and a computer to sit there and highlight. So yeah, it was the video. I was just gonna go ahead and skip it. But again, kind of this holistic viewpoint, and I wish it had a turkey here because we're switching from chickens to turkeys, but the idea of trying to understand now how the environment contributes to that behavior, and then what are the underpinnings of the genetic host potentially, because we can run different genetics through our facilities. So with that, there's lots of folks to thank through several of these funding opportunities, and several people who've either helped collect bird data or video data or counted video data. So at this point, I think that is my time, but I'll take questions as available.
questions for them. Very nice talk about share the state of art of the new facility doing lots of wonderful things.
I'm just curious, I asked your partner in crime, he wasn't sure. Do you keep genetic samples from all the birds that go through so you can go back with your phenotypes and genotype, et cetera?
Not right now, but we do have some tissues and some serum from certain birds that we've collected data on, but not all the birds.
Can you get, just like chicken, can you get DNA out of a feather? Oh,
We should be able to.
thank you.
Mhm. Oh, now I see him back there. He was hiding. I thought he was over there, but he switched places.
Any other question for them?
It's a post meal coma. Everybody's just like, I'm here, but I want to eat a little bit more maybe, or sleep, take a nap.
So Don, I have a quick question. So you talk about those microorganisms, so you find between the cage and the kind of cage-free system. Are these specific bacteria you are able to repeat it in the separate experiment?
So we did a study where we were at, let me repeat the question. So the question, sorry, I forgot to repeat the other question. So this question is, are we able to repeat seeing some of these microorganisms that change between cage-free and conventional cage? So we were doing a study where we looked at commercial hens, and then we actually did one at the Iowa State Research Farm. So we were able to replicate several of the different microorganisms in terms of like the large classes. So the lactobacillus, and lack of seeing some of the pathogenic types of organisms. But when we get down to some of those fine tune, we're not able to see that same detail. Part of it could be that we were changing from commercial facilities into a research facility, and there's some different dynamics within that.
Thanks.
Thank you.
Okay, you have
No, you're fine.
Okay, one question.
As long as it's not, is the endoplasmic reticulum the same thing as a sarcoplasmic reticulum in muscle, we're okay.
Okay.
That was my question this morning. I had kept having students ask.
Okay. Actually, this is Sue
Mm.
in a sense, and here's age showing perhaps, not yours, mine. I have this idea that with the layer study that Mike Persia did as part of our grant, you actually did deep sequencing to try and get to the species level of the microbiome. Am I remembering that correctly?
But you did, and John is working on that analysis.
It's still in progress.
That's correct.
Coming to theses near you.
Yes. So I was just wondering if you were going to apply that. It's a work in progress. Okay.
Sounds like we have much to talk about.
Yes.
Okay, thanks, Don.
Thank you.
Very much.